Student and anyone who has a curiosity or interest or is taking clinical microbiology. My name is Dr. Mayambala. I'm an instructor of microbiology and clinical lab in a local college. This is the third of several videos that I'm posting. This video is going to cover an overview of the whole field of microbiology before I get into the specific area that I plan to target over the next few videos, which is clinical or medical microbiology, because most of my students are usually nursing majors or clinical lab majors, and this is the required course. And the reason I have to specifically mention clinical microbiology is that the whole field of microbiology is so huge, it's impossible to cover all aspects of it. I'll touch on the other fields covered under microbiology very briefly so you are aware of how big this field is before I delve into the specific area of clinical microbiology. So if you follow along with these PowerPoint slides I posted, this first slide is asking the question exactly what is microbiology. I kind of briefly covered this in the first video that I posted. Basically, as the word says, micro, it's the study of living things that are small, too small to see with the naked eye. This means you need a microscope in order to look at them, in order to observe them. And again, of course, you're studying living organisms. Biology is living organisms, but they're so small, you need a microscope. This also means, of course, that one of the next videos that I'm going to post will cover several microscope types that are available and those that are specifically used in a clinical, an average clinical lab, as opposed to a specialized lab where you need higher, more sophisticated microscopes. Okay, on this slide, I also discuss, what is included under microbes. Microbes are those little living things that are so small. So they fall into 5 major groups, and most students understand that there are 5 major categories of microbes, especially those that are going to be in medical microbiology. In other words, those are involved in causing disease. They'll fall under bacteria, fungi, protozoa, and viruses, and parasites. And again, these organisms are very simple. They don't have a lot of differentiation, but again, we'll look into that because this point alone is going to be very important in understanding how we identify them and how the doctor decides what medication to prescribe to treat infections that they cause. So we'll move on to slide number 2. And again, this slide talks about two major types of cells involved in among microbes, the prokaryotic cells and the eukaryotic cells. And again, 2 types are going to play a key role in how the infections that they cause are treated by a doctor, because again, the 2 types are determined by the type of structures that they have. Number 1, the prokaryotic cells means that those cells do not have a membrane around the nucleus. If you took a course called Human Anatomy and Physiology, at the very beginning, we cover the structure of a human cell. And that human cell, we look at, the internal organelles, the nucleus being one of those. And then, of course, you have many other structures. But in the human cell, you would usually have a nucleus around it, where these prokaryotic cells, these prokaryotic microbes do not have a membrane around the nucleus. You should know that the nucleus is where you find the genetic material, DNA or RNA, depending on the type of organism it is. But that genetic material does not have a membrane around it when you're looking at prokaryotic cells. That's a very, very crucial factor. And the other factor that is unique to prokaryotic cells, that they really are very simple. They don't have a lot of structures, what we call organelles. They have just the plasma membrane, of course, which can which is the little sac that holds everything, the cytoplasm, ribosomes, and that genetic material floating. Really, no membrane around that genetic material, and only ribosomes making proteins. And basically that's it. So you can see how they would depend on whoever they infect in order to do most of their carry on most of their metabolism. On the other hand, eukaryotic cells, whether they are microbes or they are larger organisms like us human beings, they do have a membrane around that genetic material in the nucleus. So whether they have DNA or RNA, it's going to be in an enclosed nucleus, and they have a more complex set of structures or organelles in their cytoplasm. That means just like us humans, they will have most of the organelles like ribosomes and mitochondria, Golgi apparatus, and most of the other structures that we find in the human being. Again, these two categories do play a big role and implications in how infections that these microbes cause can be treated. So the eukaryotic cells, as we'll see later on when we talk about antibiotics or antimicrobial therapies, the eukaryotic cells, if they have a similar structure to similar to us human beings, that can also cause complications, because when they're choosing medications for them, they have to be very careful that the medication will kill them but not kill the human cells. Since their cells are eukaryotic just like ours, that medication would target both. So that becomes a delicate balance. You can see again how the structure of these microbes is going to play a role in infectious disease as well as infectious disease treatment. This third slide again is just giving you the general groups, types of microorganisms we're thinking of when we're looking at microbiology, especially when we're talking about infectious or medical or clinical microbiology. Of course, we have the bacteria, we have fungi, protozoa, algae, viruses, and multicellular parasites. Now, I'll make a comment about algae. Algae really do not, most of the algae are really not going to fall under pathogenic organisms, but I'll get into that in the next few slides. So in slide number 4, we've had each of those categories. The first category would be the bacteria. So those would be eukaryotic, have the features that I just mentioned earlier on. Again, they're single celled. They have unique structures called peptidoglycan walls. Peptidoglycan wall we're going to look at extensively in future videos, because that plays a role in how well they can be treated or what antibiotic can be used to treat those particular types of bacteria, because of the unique characteristics of the cell wall. They divide by binary fission, meaning they just split down the line into 2s and then 4s and so forth, and the other characteristics mentioned. Now, then the next group of organisms are the fungi. Those, on the other hand, unlike bacteria, they are eukaryotic. So again, think back on the previous slide where I discussed the two types, prokaryotic versus eukaryotic. The fungi, meaning that their cells are more like human cells, which means, treating them, you'd have to take into consideration that they're very similar to human cells because they both have a distinct nucleus. And again, the other structures described there, they have a chitin wall, they can absorb organic chemicals for energy. And of course, the most common fungi are the molds and mushrooms. But there are other fungi that cause disease like yeast and other yeast infections that you can think of, ringworm and so forth, nail fungus, things like that. Slide number 5 is talking about the next group of microorganisms, the protozoa. These are also eukaryotic, so they also have a distinct nucleus. And again, think in terms of what I mentioned earlier on. When we say eukaryotic, that means they have very similar structures to the human cell, and that means you have to take into account that similarity when considering treatment. Along with that are the algae, and again, in algae I mentioned earlier on, they're eukaryotic. Most of the algae really do not cause disease, but if they do, it's because they are they're producing toxins, algal toxins, which directly attack the human tissue and destroy that tissue. But they're not very common. So for purposes of our clarity, they're probably not going to be extensively covered. This is probably the last time that I would mention algae under clinical microbiology. But there are rare occasions where they can cause disease in cases. All right. Here comes slide number 6. This is crucial, crucial, crucial. Viruses, the category of viruses. Now, in this case, viruses are not even considered a complete cell. They're almost like little packets of genetic material. And that genetic material can be either DNA or RNA. Many viruses are RNA, but there are few viruses that are DNA. An example is HPV, the herpes, human papilloma virus. It's a DNA virus, but many others, including HCV, HIV, and COVID, those are all RNA viruses. Basically, they're just little packets of that genetic material enclosed in a little shell. It's almost like, think of an eggshell, and then inside that eggshell you have just the DNA or RNA, depending on the virus. But again, now if we know, even though they're not complete cells, we know the kind of danger they can cause because of what is, as the slide says, the coat may be enclosed with a lipid envelope, but they also have a protein on that coat at the envelope. And that protein on the envelope is what allows them to enter a cell and cause infection. And of course, along with viruses, animal parasites, again, those are also eukaryotic. Again, that means that whatever they have is very similar to human beings. Now, many human parasites, including worms, different types of worms, flatworms, roundworms, hookworms, many of them you can see, but they have some stages that are microscopic. In other words, the eggs, when they're laying their eggs, those eggs may not be, visible to the naked eye. You need a microscope. So that is one word, I guess, included in the term microorganism, because their egg stage, some of their life cycle stages, you have to look under the microscope. And in fact, in a clinical lab, when a patient comes in and they suspect a parasite, if they don't see anything as visible as that foot, a person with a worm on their foot, they will ask for a stool sample, because they want to look for eggs in the stool sample to see what stages, if the organism is laying eggs. And that's not a very popular test in the clinical lab. When you're looking over on parasites in a stool sample, you can imagine it's not a very pleasant test to do in the lab, but it is necessary in order to look for the eggs of a parasite, a suspected parasite. Okay. Now, there's this other group called the archaic, and the only reason I include this group is to make you aware that there are certain organisms that belong to this group. What is unique about them is that they exist under unique conditions, but just to make you aware that, for example, that they do exist, and the reason I mention this is that some of them exist under extreme high concentrations of salt. Those would be the halophiles, and those would exist in areas where you wouldn't expect many other organisms. Others exist in extreme temperatures. For example, you would think places that in deserts, like the Arizona desert or Sahara desert, you might not expect too much life living there, but there are certain organisms that could still exist under those conditions, and they're called thermophiles. So just to be aware of those organisms. Also, there's some methanogens. Those are organisms that produce a lot of methane gas. But again, this is just to mention them to make you aware they also fall in the category of microorganisms. And some of them can play a role in clinical microbiology, but not as significantly as the other organisms that I just mentioned in the previous slides. Now, these next two slides, next three slides, I'm just going to go through why we study microorganisms. Okay, now, and again, this is just an overall exposure before we narrow down our concentration on clinical microbiology. Now, as you can see, there are organisms all around us. They're found everywhere, even though they're small. So we need to be aware of them existing alongside us and the role they play in our lives. And some of those roles are, for example, they are used to decompose organic waste, which we think because when they collect our trash or when you flush your toilet, that waste goes, something happens to that waste to decompose the material, and it can be a good thing because it can reduce some of the toxins. They can generate oxygen by photosynthesis, in the air around us. Now, this other role of microorganisms, for example, the production of products like ethanol, acetone, and vitamins, is very important. Yes, because, for example, ethanol, the alcohol, the booze you drink, yes, one of those organisms used in the production of ethanol, for example, is yeast. We just said yeast is one of those fungi, in the category of fungi. Of course, making bread is another one. Making bread, you use yeast. Acid and vitamins would, if you cover human anatomy and physiology in the digestive system, there's certain organisms in our large intestine that play a role in producing certain vitamins. Vitamin K, biotin, and that is again that highlights the importance of those microorganisms in our environment. They're used in the production of food such as vinegar, cheese, bread. I already mentioned that. They're used in the production of products like industrial cellulose enzymes. Now, that enzyme is involved in the production of certain textiles. So again, you can see how in manufacturing microorganisms come into play. And now then, the other important role of those microorganisms, they're used in the production of disease therapeutics. Insulin is an example of that. And we'll look at many other antibiotics and antimicrobials that are produced by microorganisms. But of course, first and foremost, our first interest in this course is to understand how these microorganisms cause disease in plants, but for us, more specifically, if you're going to be a nurse, probably more specifically how they cause disease in human beings. Because until you understand how they cause disease, you cannot easily understand how to prevent them from causing that disease. So that becomes very important for us to understand these organisms, which is exactly what is being done with COVID. They had to understand how the organism, how the virus causes disease. They had to understand the structure of the virus, and then they had to understand how it causes disease in order to start to put together certain ways of preventing the virus from causing disease. And that is what research is all about. First of all, understanding the physiology of the anatomy and physiology of the microorganism, the pathogen, and then understanding how to prevent it from causing that infection. This next slide, again, is just giving many different aspects of microbiology. I highlighted medical microbiology and public health microbiology. Those are the areas I'm going to concentrate on in the next few videos that I post, because those are the ones most directly related to any student who's going to be pursuing an allied health career, like a nurse or clinical lab scientist. But you need to remember that there are many other different types of microbiology. You can use microbiology to study the ecology. You can call it ecological microbiology, agricultural microbiology. If you're going to be working in the agricultural field, you want to understand how microbes impact the agricultural discipline. Food microbiology, you're going to be working in the food industry. You need to understand how microorganisms play a role in food production and food safety, as well as other fields. And finally, not to leave out other aspects of microbiology, industrial microbiology. Again, I briefly mentioned how microorganisms can be used in production of things like products like alcohol, even clothing, as well as antibiotics, as well as certain foods, cheese and bread. So that is an aspect of industrial microbiology. And now latest, microorganisms are being used in microbiogenetics and microbial genomics. So you can see all these fields or different branches of microbiology are, so they're all different aspects, but it's impossible to all cover them at once. So one has to narrow down to what you want to concentrate on. And that is why in the next few videos, I'm going to target mostly medical or clinical microbiology alongside public health microbiology, because those two areas work hand in hand, side by side. Okay, until next time, this is the last video again. Each time I post a video, if you have any questions, comments, concerns, you can email me at KO, the letter S as in Sarah, the number 52 at gmail.com. And I'll try to address your questions or concerns or issues. Any questions, again, email me at that email address.